Hi, class, and welcome to part C of chapter 18, where we'll be looking more at the blood vessels in the cardiovascular system. Specifically, we'll start by talking a little bit more about the control of blood flow. Tissue perfusion is the actual definition of blood flow through the body tissues that's involved in the delivery of oxygen and nutrients to, and the removal of waste from tissue cells. Gas exchange occurs in the lungs, and absorption of nutrients is a type of tissue perfusion that occurs in the digestive tract. Everything that you eat, all the nutrients get absorbed into the bloodstream in your GI tract. And then urine formation, tissue perfusion, also occurs in the kidneys. And the rate of flow is precisely right in order to provide proper function to that tissue or organ. The rate of blood flow is controlled by both extrinsic and intrinsic factors. Extrinsic controls the sympathetic nervous system and hormones that control the blood flow through the whole body, and they act on our arterial smooth muscle to reduce flow to regions that need it the least. And intrinsic control is autoregulation or local control of blood flow, and this is where blood flow is adjusted locally to meet specific tissues requirements. Usually this is done by local arterioles that feed into capillaries. They can undergo modifications of their diameters, and organs can regulate their own blood flow by varying the resistance of their own arterioles. So this is just a review of intrinsic controls versus extrinsic controls and examples where both control mechanisms occur, and some resources that you can look in your textbook for: stroke volume in the heart, arterial diameter, and glomerular filtration in the kidneys. So one example is the redistribution of blood during exercise. At rest, your skeletal muscles receive about 20% of total blood in the body, but during exercise, your skeletal muscles can receive over 70% of the blood. And that's because your muscles, when exercising, need blood because they need the oxygen to contract to create ATP. Intrinsic controls the skeletal muscle arterioles will dilate, increasing blood flow to the muscle itself, and extrinsic controls decrease blood flow to other organs like your kidneys and GI tract organs to try to push blood toward your skeletal system. So here's just a look at the distribution of blood flow at rest and then the redistribution of it during strenuous exercise. And you can see how more blood goes to your skeletal muscles during exercise. Autoregulation has to do with local conditions that regulate blood flow to that area. Reactive hyperemia is increased blood flow to an area due to an intrinsic factor. And there are two types of intrinsic mechanisms, and they both determine the final autoregulatory response, whether metabolic or a myogenic control. A metabolic control's increase in tissue metabolic activities will result in declining levels of oxygen and increasing levels of metabolic products. And the effects of change in levels of local chemicals can cause direct relaxation of arterioles and relaxation of precapillary sphincters. And they can cause the release of nitric oxide, which is a powerful vasodilator by your endothelial cells. A metabolic control has to do with endothelins, which are released from your endothelium. They are potent vasoconstrictors. So we have your nitric oxide and endothelins. They're usually balanced unless blood flow is inadequate, in which case nitric oxide wins, causing vasodilation. Other inflammatory chemicals can also cause vasodilation. And then a myogenic control, any myogenic response is when local vascular smooth muscle will respond to any change in mean arterial pressure to keep perfusion constant to avoid any damage to the tissue. So we can get a passive stretched where we have increased arterial pressure stretches the vessel wall more than normal, and reduced stretched where decreased mean arterial pressure causes less stretch than normal. A long-term autoregulation occurs when short-term autoregulation cannot meet tissue nutrient requirements. So we have a long-term autoregulation that could take weeks or months to increase the blood supply. The number of vessels to a region will increase and existing vessels will enlarge. And this is really common in your heart if one of your coronary vessels is occluded or blocked, or throughout the body in people in high altitude areas. So here's a look at just the intrinsic and extrinsic control of arterial smooth muscle in the systemic circulation and what can cause your arterioles to dilate or constrict. Blood flow in special areas. So we have skeletal muscles. The blood flow varies with the fiber type and activity. At rest, your myogenic and neural mechanisms will predominate, and they maintain a flow at about 1 liter per minute to all your skeletal muscles. But in active or exercise hyperemia, during muscle activity, blood flow will increase in direct proportion to metabolic activity. So your local controls will override the sympathetic vasoconstriction, and flow can increase up to 10 times. So here's just a look at how your muscles and blood flow changes in active hyperemia or exercise, and how the oxygen, carbon dioxide, and hydrogen levels will change. To the brain, blood flow to the brain must be constant because your neurons are intolerant of ischemia. Flow averages about 750 milliliters per minute. And control mechanisms are metabolic control. So if you have any decrease in pH or increased carbon dioxide levels, these will cause marked vasodilation. Very high carbon dioxide levels will depress autoregulatory mechanisms. More control mechanisms are myogenic controls. So the decreased mean arterial pressure causes cerebral vessels to dilate, and increased mean arterial pressure causes the cerebral vessels to constrict. The brain is very vulnerable under extreme systemic pressure changes. Anything below 60 millimeters can cause fainting, or above 160 milliliters can cause cerebral edema or swelling. To the skin, the functions of blood through the skin are several. Your blood to the skin supplies nutrients to the cells, which is autoregulated in response to oxygen needs. It helps regulate body temperature. This is neurally controlled, and it's an important function of skin, and it provides a blood reservoir, which is also neurally controlled as well. The skin, blood flow through venous plexuses below the skin surfaces regulates your body temperature, and the flow varies from 50 milliliters per minute to 2,500 milliliters per minute, depending on the body temperature. And the flow is controlled by the sympathetic nervous system reflexes, and these reflexes are initiated by temperature receptors and central nervous system. So for example, as temperature rises, such as from heat exposure, fever, or exercise, you have hypothalamic signals in your brain that reduce vasomotor stimulation of your skin vessels. And this causes dilation. So warm blood flushes into the capillary beds to try to get heat to radiate from your skin. And as temperature decreases, blood is shunted to deeper, more vital organs. Superficial skin vessels can constrict strongly, and blood in the vessels may become trapped, causing rosy cheeks in the cold, for example. Your lungs, your pulmonary circuit is a little more unusual. The pathway is a lot shorter. You have arteries and arterioles are more like veins and venules, which are very thin-walled with large lumens. Arterial resistance and pressure are much lower than in the systemic circuit. And you have autoregulatory mechanisms that are opposite, where low oxygen levels cause vasoconstriction and high levels promote vasodilation. And this just allows blood to flow to oxygen-rich areas of the lung. In the heart, blood flow through the heart is influenced by aortic pressures and ventricular pumping. During ventricular systole, coronary vessels are compressed, the myocardial blood flow ceases, and stored myoglobin supplies are sufficient oxygen. And during diastole, the high aortic pressure forces blood through the coronary circulation. At rest, coronary blood flow is about 250 milliliters per minute. This is controlled by myogenic mechanisms. But during exercise, like what you could probably expect, your coronary vessels will dilate, blood flow may increase. And this is extremely important because your cardiac cells use about 65% of oxygen delivered. Other cells only use about 25% of delivered oxygen. And increasing coronary blood flow is the only way to provide more oxygen. So now we'll talk a little bit about capillary exchange. Velocity of blood flow has to change as blood travels through the systemic circulation. It's fastest in the aorta right when it gets ejected, and slowest in the capillaries, and then it increases again in the veins. The speed is always inversely related to total cross-sectional area The capillaries have the largest area, so the slowest flow. The slow capillary flow allows for adequate time for exchange between blood and tissues. So this is just a look at how blood flow velocity changes as it travels through your vessels. So velocity of blood flow is highest in the aorta, then it decreases, and lowest in the capillaries, and then increases a little bit more as it travels back to the heart. Vasomotion is the intermittent flow of blood through your capillaries, and this is due to the on and off opening and closing of precapillary sphincters. Many molecules will pass by diffusion between the blood and interstitial fluid. They move down their concentration gradients, and molecules use 4 different routes to cross capillary. They can diffuse directly through endothelial membranes. They can pass through clefts or spaces. They can pass through fenestrations, or they can pass through by active transport via some sort of vesicle. So this is just a look at capillary transport mechanisms and different ways that oxygen, carbon dioxide pass through these capillaries and the different holes and vessels you can see. Here's another look at different types of movement: diffusion directly through the plasma membrane, movement through clefts, which are spaces, movement through fenestrations, which are holes, and via transport vesicles. Fluid is always forced out of clefts of capillaries at the arterial end, and most returns to the blood at the venous end. Bulk fluid flow across capillary walls causes continuous mixing of your fluid between plasma and the interstitial fluid. This maintains the interstitial environment, and the direction and amount of fluid flow depends on 2 opposing forces: hydrostatic pressures and colloid osmotic pressures. Hydrostatic pressure is the force exerted by a fluid pressing against the wall, and there's 2 types, whether it's coming from the capillary or the interstitial fluid. So that's hydrostatic pressure. And then colloid osmotic pressure has to do with the sucking pressure created by non-diffusible plasma proteins pulling water back into the capillary. So we have different pressures that affect fluid movement. This is a look at the hydrostatic osmotic pressure interactions. I won't ask you guys to know about this, but it is interesting to study how things flow out of the capillary beds and back into the capillaries. This explains a little bit more the bulk flow across capillary walls. Hydrostatic pressure is the pushing pressure, and osmotic pressure is kind of the sucking pressure, if that makes sense. Here's also the bulk flow across capillary walls, showing the differences in pressure and how they're forcing things into and out of the capillary walls. Looking at the arterial end of the capillary versus the venous end of the capillary and how that changes. Edema is an abnormal increase in the amount of interstitial fluid. So edema means swelling, and this is caused by either an increase in the outward pressure or a decrease in the inward pressure. And an increase in the capillary hydrostatic pressure accelerates fluid loss from the blood, and this could result from an incompetent venous valve, localized blood vessel blockage, congestive heart failure, or high blood volume. An increase in interstitial fluid osmotic pressure can result from an inflammatory response as well, and a decrease in capillary colloid osmotic pressure will hinder fluid return to the blood. Edema can also be caused by any sort of decreased drainage. So excess interstitial fluid in the subcutaneous tissues usually causes what we call pitting edema. Edema is bad because it can impair your tissue function as a result of increased distance for diffusion of gases, nutrients, and wastes between blood and cells, and slow fluid losses can be compensated for by renal mechanisms, but rapid onset may have serious effects on the circulation. And this is what pitting edema looks like. So if you push into the area of edema or swelling, when you remove it, the indentation is still visible by the swollen foot. The vascular system then consists of 2 main circulations: pulmonary circulation and systemic circulation. Pulmonary is the loop that goes to the lungs and back to the heart, and systemic circulation is the much longer loop that goes to all parts of the body and back to the heart. The heart pumps blood out to the system via a single systemic artery, the aorta, And the blood returning to the heart is delivered via terminal systemic veins, the superior and inferior vena cava, as well as the coronary sinus. So this is just a review on the pulmonary circulation, deoxygenated blood going to the lungs, picking up oxygen, back to the oxygen inside of the heart. And this is a look at pulmonary circulation. Blood travels to kind of surround the alveoli, which are filled with air, and the blood capillaries that surround the alveoli are what will take in oxygen and also diffuse out carbon dioxide as waste. The important differences between the systemic arteries and veins are that the arteries always run deep, whereas veins are both deep and superficial. And you can read that explanation there. Venous pathways are much more interconnected. The brain and digestive systems have a unique venous drainage system. The brain contains what we call dural venous sinuses, and the venous system of your digestive system drains into the hepatic portal system, which perfuses through the liver before returning to the heart. So this is a look at the schematic flow chart showing an overview of systemic circulation taking into effect the digestive system and the brain system as well. Diagrams of oxygen-rich blood are shown in red, whereas oxygen-poor blood are always shown in blue. And these flow charts, one that looks like that, are called pipe charts because they're showing vessels that are closer to the surface in bright colors, and whereas vessels that are deeper are shown in darker shades. And tips for memorizing vessels will be the name of the vessel usually reflects the body region being traversed or traveling through, or the organ served, or the bone being followed. I won't ask you guys too many names of specific arteries and veins, and they're all shown here, and they give you a couple more pictures because I'm hoping you guys covered a lot of this in anatomy. So we're going to kind of skip over all the names, maybe knowing a general understanding of if I tell you where's the cerebral artery and what part of the body that would be referring to. But this goes through all the arteries. It's a great review for anatomy if you're thinking about that, but I won't ask you specifics of every single artery and vein in the circulatory system because we don't have to do anatomy again. But I think there's one more part to this PowerPoint I want to get through, and maybe there's not. Maybe it just reviews the arteries and veins. So that is the end of this PowerPoint, and thank you for listening. This is the end of chapter 18. So you'll have another lecture exam after this chapter, and then we just have one more month of school left.